My name is Deborah Wiley, and I chair the Wiley Foundation that brings you this lecture today. And I would like to take the opportunity to welcome you to the 18th Annual Wiley Prize, in Biomedical Science Award as well as lecture, which is kindly hosted here at Rockefeller University as part of their Friday afternoon series. Over the course of our 217-year history, Wiley has enjoyed a productive and successful partnership with the scientific community for the advancement of research. We conceived the Wiley Prize in Biomedical Sciences as a way to recognize and to give back to that community. Wiley chose to focus on biomedicine because it's an area continuing to experience significant progress and change. Our goal is to recognize specific contributions or a series of contributions that have opened new fields of research or advanced novel concepts or their applications in a particular discipline and demonstrate the nominees' significant leadership in the field. This year's winners, Svante Pääbo and David Reich, have indeed made such a contribution. Their work has focused on revealing the origin and ancestry of contemporary humans and our diverse populations through investigation into the genomes of modern humans, Neanderthals, and chimpanzees, allowing us to fully appreciate both our common ancestries and our discriminating differences. Our jury consists of Titia DeLonga as chair of the jury, Kais Alicati, Wayne Hendrickson, both of Columbia University, Joan Stites of Yale, and Robert Horvitz of MIT, and your own Rick Lifton, who was a Wiley Prize winner in 2008, who collaborated to make the selection of this year's award recipients. Following our award presentation, Rick will make introductions. There will be a Q&A following the presentations by our award winners. So I'd like to ask Svante and David to come up to receive their presentation award.
Thank you. Well, I am absolutely delighted to add my congratulations to Svante and David for their remarkable work, and to thank the Wiley Foundation for their generous support of this award. This was a really terrific body of work that the jury is very proud to present the award to today. Prior to Svante Pääbo's work, it was widely believed that you could not retrieve usable DNA from anything more than a couple of hundred years old, and Svante has painstakingly worked to improve the technologies, and now can go back at least, I think, to about 80,000 years ago, if not longer. And this has really transformed the capability of what we can contemplate to do from the studying of ancient DNA. And he and David Reich, who is one of the world's great analysts of small fragments of DNA and large, they've really partnered together over the last 10 years or so to really transform the field. With first the sequencing of ancient humans, and then the sequencing of really ancient Neanderthals, and then the discovery of an entirely unknown branch of the hominid tree, the Denisovans, which were first discovered from the sequencing of a finger bone that gave an unexpected sequence. And these are not just dry adds to the evolutionary tree. They instead have told us that early Homo sapiens migrating out of Africa interbred with Neanderthals and Denisovans and fragments of their genomes residing in us today. And these aren't just random bits of DNA in total. Some of them turn out to have been under strong positive selection and likely conferred particular resistance to infectious agents or acclimation to environments that may not have been survivable without the acquisition of these sequences. So it's been a spectacularly interesting and very important series of discoveries that they've made, and the jury was just so impressed with the work and happy to award the prize, this year's Wiley Prize, to them. So, getting to the business of the presentations, Svante Pääbo was a native of Stockholm. He did his undergraduate work at the University of Uppsala, started medical degree research, and then long love for archaeology turned him back to doing basic science. He ultimately received his PhD from Uppsala. And then in an interesting series of events, ended up doing a postdoctoral fellowship with Alan Wilson, a great evolutionary biologist at Berkeley, and then returned to Europe, first to Uppsala and then to the University of Munich. And since 1997, he has been director of the Max Planck Institute for Molecular Archaeology in Leipzig. And now his partner in crime, if you will, David Reich. David did his undergraduate work in physics at Harvard, then went off to Oxford, where he did a PhD in statistical genetics at Oxford with David Goldstein as his advisor. Also had an attraction, a flirtation with medicine, came back to Harvard Medical School and discovered that he really wanted to pursue basic science, and went to the Whitehead Institute, where he worked on statistical analysis of genomic data with Eric Lander, and then was recruited back to the Department of Genetics at Harvard Medical School, where he now is a professor of genetics and investigator of the Howard Hughes Medical Institute. Both of these individuals have shared numerous awards, have received numerous awards. Svante has received the Breakthrough Prize, the Louis Janté Prize from Europe, and they have shared, Svante also received the Gruber Prize in Genetics, and they have shared the Dan David Prize from the University of Tel Aviv for their terrific contributions to basic science. So the run of show will be that we will first hear from Svante, then we'll hear from David, and then we'll do a question and answer session at the end. So let me welcome Svante to the podium.
Okay, so thank you very much for the introduction and for this amazing prize. Big thanks to the Wiley family, to the prize committee, and to everyone else who made this amazing day here possible. So what I wanted to start out by doing then is to just maybe reminisce back 35 years or so to when all this started, and the feeling of frustration that really was the basis of what were to follow. And that was the frustration that those of us felt back then who were interested in the evolution of genes and of genomes. And the frustration was really that we were limited to having access to only DNA sequences of genomes of extant species and organisms. But of course, evolution is a historical process. So it was very frustrating to try to indirectly infer what had happened in the past from present-day variation. So the dream was to, in at least some cases, be able to go back and actually follow molecular evolution over time. So you then needed to sort of think about, might DNA be able to survive for a few hundred or for a few thousand years at least in organic remains? And a natural place to start seemed to be ancient Egyptian mummies, which are 2,000 to 5,000 years old or so, and look macroscopically very well preserved. So started out with was to then look histologically in tissues. So for example, from this mummy that's 2,700 years old, in the skin, in the upper layers of the skin, one could see structures that looked like cell nuclei. And indeed, if one stained this section then for DNA, you could also see that DNA seemed to be preserved in the nuclei here. And around that time, I think in '84, Alan Wilson then at Berkeley and his postdoc Rassi Gucci published some DNA sequences from the 100-year-old skin of a quagga, an extinct form of a zebra. So I, of course, also went on then and cloned DNA from this Egyptian mummy and published it even, and to great fanfare in Nature. And then over the next few years, it became quite clear that although DNA is clearly preserved in the cell nuclei here, the sequences I had published were clearly sequences from myself, from a museum curator, or something else. Because what became clear with time was that DNA in old remains are always degraded to very small size, and that happens rapidly after death. So already things that are a few hundred years old are degraded to an average size of 50 to 100 nucleotides, and even more so older things. So when you could retrieve human DNA sequences that seemed to be ancient, they were invariably very old. So in a more general sense, and the sort of technological challenge that became clear was that, first of all, if you look in ancient DNA comparing to fresh DNA, it's very little of it there, often 100,000, a millionfold less than it would be in present-day tissues. It's degraded to short size, and it carries chemical modifications. And it's present in the vast excess of microbial DNA that dominates your extracts. So as a result of this minute amounts of contaminating modern DNA that doesn't at all influence your experiments, if you work with present-day DNA, may totally overwhelm your experiments when you work with ancient materials. So over the years, this resulted in a sort of increasing mania of cleanliness, where you have separate special labs, dressed up in funny clothes, and so on, and worked out all the sort of technical issues with this. And quite rewardingly, sort of in the 90s, it became many groups that have published phylogenetic studies of now extinct animals. This was an exhibition in the Smithsonian of species, extinct species from which DNA had been retrieved already some 20 years ago. It has also become clear that you can retrieve DNA from many sources of old material, not only bones, but from things like dental calculus, from sediment samples, even from parchments, from coprolites that you find in cave sites and so on. But our big obsession, if you like, are then Neanderthals. And as you may know, Neanderthals, to the left here, appear in the fossil record something like 450,000 years ago in Europe and Western Asia, and exist in that area until 40,000 years ago or so, when they are replaced with modern humans. And you may then ask, why should we be so obsessed with Neanderthals? And to me, there's one big reason for that, really, and that is that Neanderthals are our closest evolutionary relatives to all present-day humans today. So in a way, if you want to define what a modern human is, a present-day human, from a biological or genetic perspective, it's with Neanderthals that we should compare ourselves to find in what ways are we similar, in what ways do we differ. Another interesting thing is, of course, that Neanderthals were here quite recently, until 40,000 years ago, until maybe 3,000, 4,000 generations ago. So the question is, what happened when we met? How are we related to them, and so on? And in the 90s then, we were able, with the techniques of the time, to retrieve tiny amounts of mitochondrial DNA and show that that was quite distinct from the mitochondrial DNAs of present-day people. But the big breakthrough really came then in the beginning of the millennium with high-throughput sequencing techniques, when it became possible to use an extract of DNA from such remains, make DNA libraries, and sequence all the molecules in such libraries, and start comparing it to the human genome that then became available. So the first place from which this worked for Neanderthals is this site in Croatia, in southern Europe. And it was 3 bone pieces from 3 different Neanderthal individuals, around 45,000 years old, from which we then were able to retrieve a first coarse, rough version of the Neanderthal genome, about 1-fold genomic coverage. And one could then ask, one of the first questions was, what happened indeed when modern humans came and met Neanderthals? And when modern humans then came out of Africa and came to Europe, if they mixed with Neanderthals, what we would then expect would be that Europeans today would share more alleles, more genetic variants with Neanderthals than people in Africa where Neanderthals had never been. So with the help of David, we sort of started to analyze various aspects of this genome, the Neanderthal genome. And one very straightforward way to look at that was to say, if we compare an individual from Europe, we thought a bit about whom we should sequence from Europe to be a typical European, and we were quickly clear that it had to be a French person, so we sequenced a French person, compared it to an African individual, and looked at positions where they differ from each other in the genome and say, how often does the European person match the Neanderthal? How often does the African person match? If there were no special relationship between Neanderthals and Europeans, this should be 50-50. And to my surprise at the time, I'll say, we found significantly more matching to the European individual. Even more surprising was that if we took a person from China and compared to the African individual, we again see more matching. Although most people would agree there have never been Neanderthals in China. And in Papua New Guinea, where for sure there has never been Neanderthals, we again saw more matching. So whoever we looked at outside sub-Saharan Africa, we saw this additional matching. And that then led to this hypothesis that when modern humans left Africa, they probably encountered Neanderthals early on. There were Neanderthals in the Middle East at that time. So if these early modern humans that were to become the ancestors of everyone outside Africa mixed with Neanderthals, they could carry this Neanderthal component with them in their genome also to regions where Neanderthals had not been. To the extent then that if your genetic roots are outside Africa, somewhere between 1% and 2% of your genome come from Neanderthals. This is was the first idea about this. As you will see soon, there's more complexity to how we interacted with Neanderthals, but overall, that still holds true. What has then happened since then is that we've been able to go on and produce much better Neanderthal genomes. Part of that is due to technical developments, or really all of it is due to technical developments. One of them is, for example, a new way to make DNA libraries from such old remains. DNA is double-stranded when you extract it, of course. Normally, you will use this double-stranded DNA to add on adapters in the end, so you have something you can feed into your sequencing machines. What Matthias Meyer in the lab came up with was that we should start with separating the 2 strands, and then by single-strand ligation, add an adapter to the ends of the strand here, immobilize that on a solid support, synthesize the other strand, so that for each double-stranded molecule we have has 2 chances to end up in the library, 1 for each strand. And that turned out to be one critical advance, because often there are chemical modifications on one strand that make the molecule, make us unable to replicate the molecule. So with this and other advances, we've then been able to come from the very bad first version from 2010, where we have some parts of the genome and lack others, to a very good genome, so we have or 50-fold coverage on average of each position in the genome to which we can map such short fragments. So we now have 3 genomes of very high quality. And you can begin to look, for example, in present-day people in just one chromosome here and mark in red the fragments on this chromosome in different individuals that come from Neanderthals. You will see that different people often carry different Neanderthal fragments. On average, it's 1 or 2% of your genome. But you can then also ask how much of the Neanderthal genome can you puzzle together? Can you sort of assemble from these pieces people that walk around today? And that's in the order of 40 or maybe 50% of the Neanderthal genome is still walking around on 2 legs, so to say, in different people today. So these fragments then, you can look for genetic variants, SNPs, one associated with high LDL cholesterol levels here, for example. And you can find that some such variants then sit on long haplotypes, on long chunks of DNA of hundreds of kb sometimes, that are very similar to the Neanderthal genome. And it's really the size of these fragments that show that this is not something that comes to present-day people through the common ancestor with the Neanderthals, but really comes over quite recently by gene flow from Neanderthals, because recombination has not had a chance to make these fragments small. There were other things also that came out of, that has come out in these years. And much of that comes from this site in southern Siberia, the Nisswa Cave, where the Russian archaeologist Anatoly Derevyanko and his team excavate since many years. It's a beautiful place. And as you already heard Rick mention, one of the things they found there was a tiny little fragment of the last phalanx of a pinky of a child. They were very skilled, I think one must admit, to realize that this might be something that's of hominin origin. We were not very enthusiastic about this piece. It was so small, it actually lay around for half a year in the lab before we analyzed it. But it turned out to be so well preserved that we could generate a good genome from it. And we were very surprised to find that the genome here was not closely related to Neanderthals, not to present-day humans, but went far back to common ancestors shared with Neanderthals. To date these things, it's always complicated, depends on the mutation rate that we're not sure of, etc. But in the order of maybe 400,000 years ago or so back, did this individual share a common ancestor with Neanderthals. It was clear that this was some previously unknown or undescribed group of extinct hominins that were discovered. And after a lot of debate with our Russian colleague, we decided to call them the Denisovans, after the Nisswa Cave where they were found. And still, there are only very few remains from which are known. It's a finger bone and 3 teeth, and now a small part of the cranium from this site, where one has shown that they come from the Denisovans. But we think that they have been much more widespread in the past, because you find no traces of them in the present-day genomes of people in Western Eurasia, but in Eastern Eurasia, everybody carries a small amount of Denisovan DNA on top of the Neanderthal DNA, so to say, less than 1%. And out in Oceania, it's actually up to 5% or so of the genomes of people in Papua New Guinea or Aboriginal Australians come from Denisovans. And not only that, the Denisovans seem to have had an interesting history, even more interesting than Neanderthals as far as we can see. And that's work that has been done by Josh Akey and his group, now at Princeton, where they have looked at genome sequences from people in Papua New Guinea, and they plot on this axis here similarity in DNA fragments to our Neanderthal genome. And you see that there are these parts of the genome in Papuans, as in everyone else outside Africa, that's quite close to the Neanderthal genome. They also find here similarity to the Denisovan genome, but it's quite a bit further away from the Denisovan genome we have sequenced than the Neanderthal fragments is from the Neanderthal genome. But if they instead look in genomes from people in Japan, they find these things that are quite distant from the Denisovan genome, but also fractions of the genome there that is very close to the Denisovan genome we sequenced. So this then shows that there are two different Denisovan populations that have both contributed to the ancestors of people in Japan. And you will find this sort of component wherever you look in East Asia. So there are Denisovans, as opposed to Neanderthals, have had a substructured population of different parts of the population with different histories. So if we should summarize for a minute what we think we know about the origins of Neanderthals and Denisovans and modern humans from genomes, Neanderthals and Denisovans have some common ancestry, probably in Africa, and their ancestor, probably something that one calls Homo heidelbergensis or so, come out of Africa and evolve to what we call Neanderthals in Western Eurasia, to what we call Denisovans in Eastern Eurasia. We don't know where the sort of border between these groups have been, but we know that sometime there in southern Siberia there were Neanderthals, sometimes Denisovans. Then modern humans have an origin in Africa and spread to the Middle East, and somewhere around 60,000, 70,000 years ago mixed with Neanderthals, presumably already in the Middle East, and then start spreading out over the world. They mix probably several times with Neanderthals. They mix with Denisovans in the east, additionally so in East Asia and in Southeast Asia, spread out in the Pacific, and eventually these other forms then become extinct and live on a little bit then in people today in form of this contribution to present-day people. So what's also happening is, well, I should also say that this seems to then be a difference, if you like, between non-Africans and Africans. But of course, modern humans emerged first in Africa and of course spread across Africa too. And if they mixed with earlier forms of hominins outside Africa, it's very reasonable to think that they did so also inside Africa. And there are some indications of that in the variation of present-day Africans. So I think one will, in the next few years, realize that this happened also in Africa, actually. And we're learning more about this process of mixing with Neanderthals in a more direct way from ancient DNA now. And the first hint of that came from a site in Romania, where cavers back in 2011 found this mandible that looks like a modern human mandible. It's 40,000 years old, so it's one of the earliest modern humans in Europe that lived at a time when there were Neanderthals around. So we were, of course, very interested in if this individual had mixed with Neanderthals or not. So if you just look on one chromosome here, and for some other modern humans indicating color fragments that come from Neanderthals on this chromosome, and then look on this individual from Romania, you will find that it carries a lot more contribution. And in fact, where it's black here, we have no information, so it seems that more than half of this chromosome is solidly of Neanderthal origin. So this, of course, suggests that this individual had a close Neanderthal relative in its family history. There are 7 such big regions across the genome. So you can then show that this means that this 40,000-year-old individual from Romania had a Neanderthal ancestor, either 6, 5, or 4 generations back in its family trees. It's like 23andMe for ancient people, if you like. So it seems that then from these old different forms of gene flow between modern humans, Neanderthals, and Denisovans that we can infer from the genomes, we happen to stumble across someone who was very close to this gene flow from Neanderthals to modern humans. The other such direct indication that we recently got is from the Nisswa Cave again. And that comes from studies of the vast numbers of non-diagnostic small bone fragments that you find at archaeological excavations that are so small, so you cannot judge if they are humans or animals. The vast majority of them are, of course, from animals. So together with Tom Hyam and this group in Oxford, They have screened with mass spectrometry for collagen, where you can distinguish between animals and humans. They screened over 2,000 such bone fragments and found one, so this one, that turned out to be of hominin origin. They couldn't say what type of a hominin, but Vivian Sloan in our group then worked on that. She first looked at the mitochondrial genome. That turned out to be Neanderthal, indicated in blue here. But when she then went on and sequenced the nuclear genome and plot here in blue Neanderthal-like variants and in red Denisovan-like variants, you find that each chromosome is solidly both Neanderthal and Denisovan. So this then suggests that one of her chromosomes in each pair is of Neanderthal origin and one of Denisovan origin. So you could then show that this individual that lived about 90,000 years ago in southern Siberia had a mother that was Neanderthal because of the mitochondrial DNA and a father that was Denisovan. But not only that, so if you look carefully at this, you can find small regions like this, blown up here, where you find that this individual, for a few megabases, look just like a purebred Neanderthal, if you like. So this then suggests that the Denisovan father had some Neanderthal ancestry back in his family history, but several thousands of years back. So in this scheme of things, of gene flow between these groups that we knew from studying the genomes, we then stumbled across another one of these here. So one lesson from this is, I think, that human groups have always mixed with each other, even these rather distinct groups, Neanderthals, modern humans, and so on, when they met. But they cannot have met that often because they are still distinct, if you like. And something else that sort of strikes me is then how that we have now twice been so lucky to stumble across these things. So in the Nisswa Cave, we have only 6 individuals for which we have genomic information. One of them turns out to be this first generation offspring of a Neanderthal and Denisovan. If we look at modern humans that are so old, that lived at a time when there were Neanderthals around, we still only have 3 individuals for which we have genomic information. One in Western Siberia here, this Romanian individual, and this individual outside China. And one of them then turns out to have a close Neanderthal ancestor. So we seem to be too lucky. I mean, these are small numbers. It's hard to make a big story out of it. But I would suggest that this then means that it might be that part of the story, at least, about how Neanderthals and the Denisovans disappeared may simply have been that they were absorbed into modern human populations that were probably more numerous, they mixed with them, and therefore lives on in present day people. I think it could have been different histories of how these interactions happen in different parts of the world at different times, but this may be part of it. Then, for the last minutes here, I would like to then ask a question: what does this mean anything functionally for people today, this contribution from these now extinct groups? I just want to give a few examples of that then. One of the first examples that surfaced was a variant that exists as SNPs that exist, and you will find them in this typical pattern of existing outside Africa, not inside Africa, and particularly in this case in Asia, where up to 25% of people carry a variant of a lipid transporter that has 4 amino acid differences between this variant contributed from Neanderthals and the more common variant. And this variant is associated then with increased risk for type 2 diabetes. So if you make a tree of these things, the risk alleles is clearly very close to the Neanderthal genomes, actually both Neanderthal genomes we had, and the protective alleles, the modern allele, if you like, is quite distinct from that. This is an example of where we actually begin to learn something, I think, slowly about the physiology of Neanderthals, because there's a lot of work from the medical literature where you learn about these risk variants, and when we now learn that these are actually the Neanderthal variants, we begin to learn something slowly about the physiology of Neanderthals. You may also speculate about why a variant that increases your risk for diabetes have increased in frequency as if it was positively selected in Asia. And I think you may speculate in this case that this may be due to that variants that give us diabetes today when we eat almost too much during our whole life may have been of an advantage in periodic starvation in the past. It may be some Neanderthal adaptation to starvation. So that's just one example of several contributions then. The Nisswans, have they contributed? Yes, they have. There are several examples of that. The most striking one may have to do with high altitude adaptation in Tibet, where people live at high altitude, very little oxygen in the air. And it was already known that there's one variant there that exists at 80% allele frequency or so in Tibet. And that's a variant of a gene called EPAS1 that has to do with how much oxygen your blood can transport without doing it by increasing the number of erythrocytes that give problems with blood clots and so on. And Rasmus Nielsen's group at Berkeley showed that this variant came into the ancestors of Tibetans from the Nisswans here. It's quite fascinating that we may not have had so large populations in the high plateau in Tibet had it not been from this contribution from the Nisswans. Some of these things seem to have been so important so that they have come over many times from these archaic groups, and both from Neanderthals and from the Nisswans. So one example of this is a cluster of toll-like receptors that again exist then outside Africa primarily. When they exist inside Africa, they seem to be associated with gene flow back from Europe to Africa. These variants clearly come both from Neanderthals and from the Nisswans here. And they are associated with higher expression of these toll-like receptors, which are important in innate immunity. So people who are homozygote for the Neanderthal variants express more of this than those that are heterozygote or homozygous for the modern variants. And this associates then with carrying these archaic variants with increased resistance to Helicobacter pylori infections, and also with an increased risk for allergic problems. So there's a picture emerging where Neanderthals and Denisovans have lived for hundreds of thousands of years in the environments outside in Eurasia, had adapted to that variant. Then comes these newcomers from Africa, mix a little bit with these individuals, and take up some variants that are advantageous that then increase to high frequency. It's something we would call adaptive introgression or so. But most of the variants that have come over from these archaic groups are associated with negative consequences, with many skin diseases, kidney diseases, hypercoagulation, and so on. There actually are indications that many of them, if you look, for example, Josh Akey's group again, has looked at allelic imbalance. If you carry one allele that's of Neanderthal origin and one that is of a modern origin, you would expect them to be expressed 50-50 in your tissues, but in one example here, the Neanderthal variant tends to be expressed in lower amounts, and that's particularly prevalent if you look in genes expressed in testicles and in the brain. The Neanderthal alleles are substantially lower expressed. We see that too if we say there are around 2% contribution of Neanderthal DNA genome-wide in Europeans, but if we look in promoters, that's significantly lower. So it seems that particular gene expression is sort of a bad thing. Something that then also now slowly becomes possible when we have 3 Neanderthal variants, 3 Neanderthal genomes, is that we can begin to look for things that seem to be typical of Neanderthals, that have changed here, and it's not in modern humans and not in apes. And one such example that we work on just these days in the lab is this ion channel, the sodium ion channel, that the postdoc in the lab works on, and his professors asked us not to spread this around. It's this ion channel here that is not just any ion channel, but it's the one that sits in peripheral nerve endings and initiates a sense of pain. So it carries 3 amino acid differences than in Neanderthals to the variant in present-day people. When he expresses these in frog oocytes and look at it electrophysiologically, it shows a decreased inactivation. So after stimulation, you have a bigger ion current going into the cells. So I'm not an expert on this at all, but these receptors, and they open on activation, and then they are inactivated, and it's in inactivation that is slower in these Neanderthal variants. So you can then show that none of the 3 amino acid changes individually is responsible for this, but the 2 intracellular ones together have a sort of epistatic interaction that explain the whole effect. So we then wondered, what does this mean really physiologically? And one great thing that is coming around in biology are these huge cohort studies. One of the biggest ones is the UK Biobank, where in this case there were 360,000 individuals that we could look at their genome and then look at if they and find that they actually 0.4% of them. So it's very rare, but there are individuals today that still carry the Neanderthal variant. And they had then answered questions. There were 19 questions in the questionnaire. So these 360,000 people had answered about pain. Have they had a headache in the last year, stomach pains, etc., etc.? So we could then sort of say, what does this sense of pain aggregated over these questions correlate with? The sad thing as you get older is that the biggest correlation you see with increasing age. The older you get, the more pain you report in your life. We could get carried away sort of and ask other things. Men and women, for example, do they differ in how much pain they report? We had different bets before they did this, and there's actually no difference, although we have quite a lot of power here to ask the question. But the carriers of this Neanderthal variant do report significantly more pain than the people who don't carry this variant. So that corresponds, if you like, in this scheme of things, to 8 years of additional pain in your life if you carry this Neanderthal variant. So, of course, these individuals are all heterozygote for this Neanderthal variant. Neanderthals were homozygote. Of course, pain sensitivity is modulated at the level of the spinal cord, and a lot is in the brain, of course. So we can't know that this means that Neanderthals really experience more pain, but people carrying these variants today, they do that. So maybe we have to sort of reconsider a little bit Neanderthals and the image we have of Neanderthals. Maybe they were not so brutish, maybe they were actually wimps. And just to add then, the question is how far back could we go with this? I just want to then show you the very oldest sequences we have. They come from these individuals, which are around 430,000 years old, found at this deep cave site in Spain, where they find many, many remains of many individuals. We worked first on the mitochondrial genome there, and just to give you a sense for how much work this is, so sort of we sequenced over 500 million DNA fragments to reconstruct the 15,000 nucleotides in the mitochondrial genome. Could then show that that is related to this mitochondrial genome of these denisovans. But when we look in the nuclear genome, and currently we have around 1% of this genome, we could show that it's mostly on the Neanderthal variants. So we now have some sort of early ancestor of Neanderthals here, where we are struggling to get more of the genome. But this sort of gives some hope, I think, that so far we've sort of not gone much beyond 100,000 years. Now we are at 400,000 in this exceptional situation there, that in some such exceptional circumstances, maybe we can go back even approaching 1 million years or so in the future. So that is one exciting direction. I think the other exciting direction that David will now talk to us about is actually looking in the Holocene in the last 10,000 years and follow evolution there. I can't mention everyone who have contributed. Many groups have contributed fossils to this, very valuable fossils. Some people are very crucial. Current in the lab, Janet Kelso, who runs all the bioinformatics, Matthias Meyer, who develops much of the molecular techniques we use, Ben Petru does the population genetics. Many people have helped us, particularly David here at the board, who will now go on and tell us more fascinating stories. Thank you very much.
Thank you. So it's really an amazing honor to be co-recipient of this prize with Svante Pääbo, who's such an extraordinary scientist in so many ways, and really a testament to the field itself, which really has grown up to be an important field really in just the last decade. And so I'm very happy to see a prize also generally for this field, which represents many people, not just the two of us. So what's really happened and why this has been such a transformative change is that what's really been invented in the last 10 years is a new scientific instrument. Svante's talked about a history of 35 years, and that's very important and deep history, but there's really been a tremendous transformation really beginning in 2010 when it became possible for the first time to generate whole sequences of ancient people, ancient animals, ancient plants. And that's turned ancient DNA from a niche field that made some modest contributions to understanding the deep past into something that was truly transformative. And so just like other scientific instruments that have been truly important in the past, this one, a measure of a new scientific instrument and how powerful it is that when you turn it to something that has not yet been looked at before with that scientific instrument, it reveals something that has never, very surprising, similar to a microscope 300 or 400 years ago when they were first turned onto the world of microbes, they revealed whole worlds never before imagined. And this is what's happening with ancient DNA now. It's revealing surprise after surprise and challenging orthodoxies that were established in other areas. So ancient DNA starts, as Svante mentioned to you, with a bone. This is an ear bone, which tends to be a particularly rich source of DNA from the side of the skull. In a clean room where the goal is to protect the bone from the person who's handling it, we drill or use another method to get beneath the surface of the bone to isolate a part that we think is particularly DNA rich and least likely to be contaminated. We turn that bone into a powder with some means or another. We then dissolve the powder in a watery solution that removes the protein content and the mineral content, and then we turn that extracted DNA into a form that can be sequenced in one of these modern sequencers. The modern sequencers are one of the series of about 4 game-changing innovations that have made whole genome sequencing of ancient remains possible. The cost of sequencing per DNA letter has dropped by a factor of literally about 1 million in the last 2 decades. And that, along with these very much more efficient means of extracting DNA that have been pioneered in Svante's lab, of turning them into sequenceable form, also pioneered in Svante's lab, and finally enriching parts of the genome for the places that are most informative about history, again pioneered in Svante's lab, have made it possible to regularly extract large amounts of data from these ancient human remains. As a result, from the first genomes published in 2010, there's been a rapid increase. So for example, since 2013, there's been a more than 100-fold increase in the number of genome-wide data sets of humans available. And that rapid exponential growth continues to increase and will go through several more doubling cycles in the next few years. And as with any instrument, for example, a microscope, when you turn up the ability to blow things up and look at small by a factor of 10, you just see things that were never seen. Every few years, we're seeing types of things that we've never seen because of the increase in sample size. So I'm going to tell you a story of 3 surprises, and the first I will barely tell you about because it was much of the subject of Svante's talk. The first surprise was that the sequencing of these archaic humans has revealed unexpected and unimagined complexity in the interaction amongst ancient humans. This is a depiction of some of the relationships between these archaic humans from a paper in 2014 between these Denisovans, the ones that interbred with people in New Guinea, for example, and the ones that are distantly related to them, the Neanderthals, interbreeding possibly from Neanderthals into the ancestors of non-Africans, unknown archaic humans who interbred with the ancestors of Denisovans, who could be reconstructed statistically from the little trace bits they've left behind in Denisovan genomes, and other interbreeding events and mixture events I haven't even talked about. So the second surprise is that white people, for example, people who are thought of as West Eurasians today, Europeans, Central Asians, Near Easterners, who today are genetically quite similar to each other. For example, on average, 10 times more similar to each other, measured by a standard measure of genetic similarity than Europeans are to East Asians. So white people are actually a recent phenomenon. So if you're able to roll back in time to 10,000 years ago, as we were able to do in 2016 when we obtained genome-wide data from people in Western Europe, who at the time were hunter-gatherers, not yet farming, from Eastern Europe, who were at time hunter-gatherers, not yet farming, from early farmers from the Levant, present-day Jordan and Israel, and early hunter-gatherers and farmers and herders from Iran. Those 4 groups in a region where today people are as genetically similar, very genetically similar to each other, were each as different from each other as Europeans and East Asians. So if you were to go back in a time machine, as you can with ancient DNA, and look at the diversity of the world 10,000 years ago, it would look profoundly different from how it does today. It would not be have the fault lines of genetic division or blurry gradients of genetic division we would see today. There would be a very different structure, which would not be very recognizable. So how did that situation get to be how it is today with a low level of differentiation? Well, we know that from ancient DNA. So if you look at individuals from about 6,000 years ago from this broad region, what happened is these 4 sources of ancestry, Western European hunter-gatherers, Eastern European hunter-gatherers, early farmers and from the Levant and Iran, none of them disappeared. These bars are bar plots showing the percentages of ancestry. They just mixed with each other. And they mixed with each other in different proportions in different parts of this region, reducing the degree of differentiation across this region by a factor of 3. And then by 4,000 years ago, the low level of differentiation seen today had been achieved through further profound mixture. So the third surprise, and what I'll spend the rest of my time talking about, is one that was really quite unexpected based on the previous investigations from archaeology and even from linguistics and other methods for interrogating the past that had been available prior to the ability to directly sequence DNA from ancient human skeletons from archaeological sites and see how they're related to each other and to people today. So the surprise is that massive expansions of people and gene flow from the Eurasian steppe, Russia, Ukraine, Central Asia, Kazakhstan, transformed both Europe and India in terms of the population structure in the last 5,000 years. And I'm going to talk to you in a little bit more detail about the evidence for that. So one of the things that's at play in genetic data is the big question, to what extent, when you see cultural change evident in one way or another from the thin evidence we have from prehistory, to what extent are profound cultural changes and events that you see propelled by movements of people? So for example, when you're at an archaeological site and digging a trench through the archaeological site, and in one layer you see pots made in a certain way, and in a deeper layer you see pots made in a completely different way, is that because a new fad swept through? Is that because people copied an idea from their neighbors who made pots in a similar way? Or is it because new people swept through and mixed with the existing group or displaced them altogether? So that was an unanswerable question prior to the ability to get DNA out of the ancient skeletons often buried in graveyards or under the houses of some of these ancient cultures, and is now answerable. And the answers that are emerging are much more often than not discrepant with the most likely or even the second most likely explanations that were suggested by many of the fields that were investigating these issues. So here I'm going to focus on Europe and South Asia for the rest of my presentation. And I'm going to show you in this map two maps of Europe showing two important pieces of evidence for cultural transformations in the past. So on the left is a map of the spread of farming into Europe. Farming wasn't invented in Europe. It was invented for the first time in the world between about 12 and 11,000 years ago in the Near East, eastern present-day Turkey, Syria, and northern Iraq. And it spread from there slowly over the region. And after 8,500 years ago, it moved explosively into Europe at the same time as it moved explosively eastward toward South Asia. So it moved across Europe, and by 6,000 years ago, it was in Britain, and about that same time in Scandinavia as well. So that's a profound cultural change, a total transformation in the economy. Was it accompanied by movements of people? Similarly, today in Europe, almost everyone, with some exceptions, speaks Indo-European languages, a closely related set of languages, which it's hard to imagine have a common ancestor more distantly than maybe 8,000 years ago or so. And it's a great deal of homogeneity compared, for example, if you look at a similar sized region of South America, the languages are almost unreconstructably different from each other. So it suggests some process in the past that radically transformed culture. Language is an important piece of evidence about culture, and that enforced a relative homogeneity of languages that then drifted into somewhat mutually unintelligible tongues since that time. So how are these events connected or not connected to movements of people or to each other? So lurking behind this is the 230-year-old problem of Indo-European language origins, which was first noticed several hundred years ago by people who noticed not just that these languages of Europe are similar to each other, but that they're also similar to languages in Iran and northern South Asia and Armenia. How did this great similarity come to be? So in 2014, we had the following situation from ancient DNA. We only had maybe a few dozen ancient genome sequences, and because of the selection of sequences that was made at that time, we didn't yet have DNA whole genome sequences after about 5,000 years ago. People first were interested in the transition from hunter-gathering to farming in Europe, and so all the sequences tended to be older. And so in this bar plot, I can show the ancestry proportions of the individuals between 5,000 and 8,000 years ago that we had data from Europe. These are each different individuals. And you see people were mostly of farmer ancestry, similar to Western Anatolian farmers that we now have a lot of data from western part of present-day Turkey, and some proportion of hunter-gatherer, typical European hunter-gatherer ancestry, and it varied in proportions in different parts of Europe. But if you do the same reconstruction today, there's something else. And the single most important part of ancestry in many present-day European populations is this red component in these bar plots that comprises up to half the ancestry of some groups in Europe today. So sometime between 5,000 years ago and today, this red stuff was injected into European populations, and it made a major impact. And so that prompted us to actually try to collect data from 5,000 years ago till about 2,000 or 3,000 years ago to see if we could catch that process and see how it had happened. So in 2012, we already had a clue not from analyzing ancient DNA, but from analyzing modern DNA. And my colleague, Nick Patterson, and I developed a new statistical test that we call the 3-population test, and I will show you a little bit about how this test works here. So this is a cartoon representation of a DNA strand, and we're looking at the same position in the DNA in 3 populations: a Native American population, a European population, Northern European population, and a Sardinian population from an isolated island to the south of Europe. And this pie chart shows the proportion of the DNA sequences from this Native American population at this location in the genome, which I've picked to be one that's variable, where some people might have the DNA letter adenine, and some people might have the DNA letter thymine, for example. It's the proportion in black of the adenine. And so you can ask the question, and it's a large sample of Native Americans, a large sample of French people, a large sample of Sardinians, does the frequency of this thymine tend to be intermediate in the French people between the Native Americans and the Sardinians? If it is, when averaged over hundreds of thousands of positions, that provides unambiguous evidence that the French people are a mixture of two groups differently related to Native Americans and Sardinians. Now, of course, it doesn't mean that it's actually those two groups that mixed, but it means groups related perhaps very distantly to them mixed. So with this statistical test in hand, we took a data set we had of about 50 populations around the world, and we took each population in turn, and we took every other pair of populations, and we used them as sources for the test population. And when we looked at Northern Europeans, for example, French, we got a huge signal of mixture. And the two strongest contributors to that mixture were, on the one hand, Sardinians, and on the other hand, Native Americans. And it was definitely Native Americans. It wasn't Siberian populations or Australian populations or New Guinean populations or South Asian populations or African populations. It was definitely Native Americans. And of course, we didn't think that Native Americans had sailed across the Atlantic or walked across the North Pole or something like this into Europe. Instead, we argued a more sensible idea that there used to be a population in the past, what we called the ancient North Eurasians, a group that had sometime lived more than 15,000 years ago in northern Eurasia, and its descendants crossed the Bering Land Bridge into the Americas to contribute to the population that became Native Americans. And some of its other descendants contributed to Europeans, mixing with people from the Near East who brought farming to form present-day Northern Europeans. So this is what one calls a ghost population, a population you can reconstruct based on the traces it's left behind in present-day people in mixed form, but that no longer exists today. It's a statistical reconstruction. And so we've seen many ghost populations from genetic analysis, and you can make a prediction that you might find them in ancient DNA. A year and a half later, this one was found. So a group working in Denmark obtained DNA from a 24,000-year-old boy buried along the shores of Lake Baikal in east-central Siberia. And this individual, as shown in this heat map, had relatedness both to Native Americans, as shown in this red color here, and to Europeans, but very little relatedness to the people who live in that part of the world today, which reflects, as we now know, post-Ice Age movements from south to north of East Asian-related people displacing the previous primary population of that region. So now I'm going to tell you through a movie, a still movie, how it is that the ancient population of Western Eurasia transformed into the modern one. So what these 1,000 or so dots are individual genome sequences or individual whole genome data sets from people from the following locations on this map of Western Eurasia and North Africa. And the data that's used to construct this plot is a table. So we're looking at about 600,000 places in the genome where people vary. Some people, for example, might have an adenine DNA letter. Some people might have a thymine DNA letter. And so the table has about 600,000 rows corresponding to each of these positions, and it has about 1,000 columns corresponding to each of the individuals. And in each cell of the table is a 0, 1, or 2, depending on whether you have 0, 1, or 2 thymines, for example. So we multiply the table by itself to see how closely related each one of these 1,000 individuals is to each other individual based on how similar the genotypes are, the DNA letters are. And then we use principal component analysis, a method for visualizing how similar individuals are in a table like this to separate the samples most efficiently. So the x-axis on this plot is the most efficient way of separating the samples, and the y-axis is the second most efficient way of separating the samples. And there's a third axis and a fourth axis, but that can't be shown on a two-dimensional plot. And if you look at the plot, you see a remarkable pattern where the European samples are all on this gradient on the left. The Near Eastern samples are nearly all on this gradient on the right. The ones in the south are nearly all on the bottom, and the ones in the north are nearly all on the top. And there's a gap in between where there's relatively too few groups today, and these tend to be groups with known, more recent, or plausible contacts between the Europe and the Near East. Now I'm going to gray out these points, and these are the modern individuals, and now I'm going to show you where the ancient individuals fall relative to the modern individuals. So here's where the ancient hunter-gatherers of Europe fall. They fall beyond Europe in the direction of European differentiation from the Near East, and the correct interpretation of this can be shown in many, many ways. This is just a qualitative impression of the data. The correct interpretation of this is that Europeans today are a mixture of people like this and of people like this, and that people like this no longer exist in unmixed form in Europe today. That's why we don't see any points on the plot that way. Beginning with the explosion of farming into Europe after 8,500 years ago, we see a pileup of individuals over here on top of present-day Sardinians. Sardinians we now think of as a relatively isolated descendant population of the first farming spread from Western Anatolia into Europe that's been little impacted by subsequent flows. Meanwhile, in the steppes north of the Black and Caspian Sea, this group forms, who is a mixture primarily of Eastern European hunter-gatherer ancestry, these green spots, and Iranian hunter-gatherer or Caucasus hunter-gatherer or farmer ancestry. They mix over here. But you still see that people who fall in the plot where Northern Europeans do had not yet formed. This happens only after 5,000 years ago in association with two widespread archaeological cultures of Europe known as the corded Ware complex and the Bell-Beaker complex that spread each one from Eastern Europe and the other from Western Europe across that subcontinent. So a summary here is that Europe has been massively impacted by two migrations in the Holocene, the period over the last 11 or 12,000 years. The first bringing farmers from the Near East, displacing about 80% of the ancestry and mixing with the local groups. And the second one, the one that wasn't expected and was very surprising, bringing pastoralists from the steppes north of the Black and Caspian Sea and bringing this red ancestry into Northern Europe and eventually into Southern Europe, as well as it percolated down through over the next few millennia. It was assumed that this archaeological phenomenon, which was known, could not have made a major demographic impact on Europe, reason being that once a densely established farming population is in place, how could it be that thinly spread herders can make an impact? In the same way, how could the British or the Mughals have made an impact on India? They barely did demographically. They politically controlled the subcontinent for hundreds of years, but almost didn't make a dent demographically on the subcontinent. But here it's profoundly different. These thinly spread, economically relatively inefficient people somehow displaced about half the ancestry of Europe. So who were these people who came from the steppes north of the Black and Caspian Sea? This is a photograph of the grave of one of the people we sequenced, an individual with an early copper mace with his head bashed in. They spread from the steppes north of the Black and Caspian Sea. They were members of an archaeological culture that achieved a tremendous transformation in lifestyle and economy in the steppe. They emerged shortly after the invention of the wheel, which was not invented by them, but was intensively used by them as it spread like wildfire over many places in Eurasia. And they had also come after the recent domestication of the horse, which potentially they were involved in doing themselves. And they took the wheels, their carts drawn by horses, into the open steppes far away from the river valleys, taking their supplies out into the open steppes, and for the first time were able to economically exploit these lands. And they expanded very dramatically, replacing the previous very disparate cultures that were there with a homogeneous type of cultural material that is seen after that point all the way from Hungary, all the way in Central Europe, all the way to the Altai Mountains on the boundary of Mongolia. So I wanted to show you briefly what happened as they got to the westernmost parts, not they, but their descendants or people who had ancestry from them of Europe. So if you look in Britain from 6,000 years ago, onward, we collected and published at the beginning of 2018 a study which included more than 150 British people from this period, from 6,000 to 3,000 years ago. And this is on the y-axis is an estimate of how much ancestry they have that ultimately comes from the steppe north of the Black and Caspian Sea. And as you see, there's none of it until about 4,500 years ago. The last big stones at Stonehenge go up in this time, built by people who were descended from the first farmers of Britain. But suddenly, about 4,400 years ago, you see these people come in, and it's about a 90% population replacement with at most 10% retention of the local group. And it's people like this who are genetically similar to people in Britain today. The builders of Stonehenge have contributed very little to the people who live in Britain today. So here's a paper we published a few weeks ago, the same type of plot, but in Iberia, which is Spain and Portugal today, the same time period and the same way of showing the data. From 6,000 years ago to 4,500 years ago, again, you see none of this ancestry from the East. And suddenly there's a few hundred-year period when they're overlapping with people with a lot of it and people with none of it. And then about 4,000 or 4,200 years ago, they collapse into each other, these two populations, and they thoroughly mix. And so it's about a 40% population replacement. Not so dramatic, but still tremendous as what happened in Britain, but there's something quite remarkable about it. So if you look at the coloring of these dots, the open circles are females and the colored circles are male. And the reason we're coloring the male circles is because this is showing Y chromosome data, which is only carried by males. And it's colored according to whether the Y chromosomes are typical of the first farmers or hunter-gatherers of Europe, that's blue, and whether they came and were unique to the steppe north of the Black and Caspian Sea, that's red. And what you see in Iberia is that after 4,000 or 3,800 years ago, there is no local Y chromosomes left. So even though it's only a 40% replacement by people from the East, it's 100% replacement on the male lineage. And that means that the incoming males succeeded in displacing the local males and contributing to subsequent generations almost completely. And it's telling you something about the nature of the interaction between the incoming people and the people who were there already. So Yamnaya ancestry also moved east. And so this is a paper that we're in the process of bringing to publication. And so this is data from more than 500 individuals from these red points here. There's India, there's Kazakhstan, and we compared them to many, many modern groups in South Asia. And I'm going to focus on just one part of this story, and that's specifically people from these sites here dating to about 4,000 years ago, who built great towns like this about 4,000 years ago and are members of an ancient civilization that was just discovered by Soviet archaeologists in the 1970s called the Bacterium Margiana archaeological complex. And so if you look in principal component analysis, the same type of plot I showed you before, what you see is something quite interesting. You see a main group of samples, which is similar to contemporary farmers of Iran. But you also see outliers, and outliers is one of the most powerful things that's coming from large sample size ancient DNA studies. So if, for example, you sample people from, say, a town like Tokyo or a city like Tokyo, and you sample 10 people, it's likely that genetically most of them or all of them will be very similar to each other. But if you sample 100 people or 1,000 people, you will begin to see people with different ancestry. And that's very informative because it's telling you the cultural groups that people there are coming into contact with. That's what's happening now with ancient DNA. We see not just a main group, but we see outliers. And they tell us a story about what happened that would not be appreciable without it. So the main group is genetically similar to ancient Iranian farmers. Before 4,000 years ago, some of the people buried in the graves were very different, and they had ancestry similar to the hunter-gatherers of the north. And that's telling you that these farmers were interacting and sometimes incorporating into their communities and burying in their cemeteries people who had ancestry from hunter-gatherers in the north. After 4,000 years ago, you see people with Yamnaya steppe pastoralist ancestry, the people I told you about before who moved their ancestry into Europe, some hundreds of years before that. And we see in multiple sites in Kazakhstan, too, it arrives just after 4,000 years ago, or just around that time. Then also around 4,000 years ago, we see this. we see people with South Asian ancestry also buried at these sites. And when we saw this, we stopped our paper for a year and a half. And the reason is because we didn't expect to see this at all. So the first individual was a girl buried in a pot from 4,000 years ago, and we studied more and more individuals from this site. We found 2 more, and then we found a second site from eastern Iran that had another 8 such individuals. So we now have 8 individuals who have a mixture of South Asian-related ancestry, similar to people in southern India, and an Iranian ancestry which is different from these people. So these are clearly immigrants from somewhere else, and we know that these sites were in cultural contact with the Indus Valley civilization, which is one of the great early civilizations of the ancient world and was contemporary to this one, but in South Asia, in present-day Pakistan, and northwestern India. And we speculate that these people are migrants from the Indus Valley civilization who were present in these great towns to their north and to their west, and were associated with the cultural materials that also are traded from that civilization. So this allows us to build a model for the formation of present-day South Asian populations, which we have data from. And we can model present-day South Asian populations quite accurately now as a mixture of 3 sources, one related to ancient Iranian farmers and hunter-gatherers, one related to southern peninsular Indian people today, one related to steppe pastoralists. And at that time, the Indus Valley group had none of that ancestry, steppe ancestry, but variable proportions of these 2 sources, indicating that, like today, it was a diverse population, but a simple gradient of 2 sources. After 4,000 years ago, we have DNA from northern Pakistan that begins to have steppe ancestry in different proportions as a people somewhere along this gradient mix with someone here, with groups here. But you still don't have groups like South Asians today. You have the formation along this gradient of one of the main source populations of South Asia, and that group mixes with another of the major source populations of South Asia, which we think forms as groups like this, mix with peninsular South Asians. After this civilization, the Indus Valley civilization declines after 3,800 years ago, they mix with the steppe people coming from the north and peninsular South Asians from the south, and mixture of these 2 mixed populations produces the gradient of present-day populations shown in blue seen today. If you look at people who fall off gradient having too much steppe ancestry, they tend to be groups who are in the Brahmin or Bhumihar groups, which are groups of traditionally priestly status in the traditional Indian social status caste system. And this provides a new line of evidence that for an Indo-European steppe contribution to Indo-European culture, because the Brahmins in northern India are the traditional custodians of the sacred texts of Hinduism that are Indo-European texts. And this is beyond the shared ancestry from Yamnaya steppe pastoralists that is shared between Europe and South Asia and is a very plausible candidate for the spread of Indo-European languages. So in summary, there's a parallelism here with this mass movement from the steppe. Farming develops 11 to 12,000 years ago in the Near East, explosively moves both west and east after 9,000 years ago, and then spreads across the ecologically complex zones of these 2 subcontinents of Eurasia. Meanwhile, in the steppe north of the Black and Caspian Sea, after 5,000 years ago, people spread both east and west, get a little bit to earlier than they do to South Asia. They mix with farmers at the periphery, and mixtures of these 2 mixed populations then form the primary gradients you see in each region today. Today in South Asia, though, population structure is much, much higher than it is in Europe, and that reflects the traditional caste system, which prevents groups from mixing with each other. And this is true whether you control for language, social status, or region of origin. And the correct way to view in many Indian groups today is not as a single large population with more than 1 billion people, even though that is the census count of India, but rather as thousands, at least 5,000 in fact, endogamous groups and many, many small populations. We can actually show from genetic data that these small populations were, some of them, founded a very, very long time ago. For example, the Vaisya from Andhra Pradesh, it's a group of about 3 or 5 million people in the area where Hyderabad is today. That group, many people in that group share ancestry from a relatively small number of founders 2,000 to 3,000 years ago. And if there had been more than 1% contribution on average into these groups from their neighbors over the last 2,000 to 3,000 years, we wouldn't be able to see that. So that's telling you that these people who live cheek by jowl next to many other groups somehow maintained strict endogamy. Some anthropologists, for example, in this book have argued that caste in modern India is an invention of colonialism in the sense that it became more rigid under colonial rule, the British using it as a way to rule and control India. But in fact, genetics shows that many of these current distinctions are very ancient and that strong endogamy must have shaped marriage patterns for thousands of years. So what's quite extraordinary with the genetics, that it's not only documenting ancestry relationships, but it's documenting cultural change. After 4,000 years ago, there's a convulsive mixture as the Indus Valley population, or the speculative one, represented by those outlier individuals, mixes both with people to the north and then people to the south, west, southeast, to form the ancestral North Indian and ancestral South Indian groups, and these groups mix in turn convulsively with each other, and then that region snaps into an endogamy between 2,000 to 3,000 years ago, and there's relatively little mixing, although surely it occurs in some places as well. So this is a big medical opportunity in South Asia. You can show that approximately 1/3 of groups in India are from groups with clinically significant founder events, a relatively small number of individuals giving rise to a large number of people today, sometimes millions of people. And so if you look and rank about 200 or almost 300 groups by the strength of their founder event and put Jewish groups and Finnish groups, groups with very strong founder events that have clinically significant panels of mutations, those are these red lines here. There's about 1/3 of groups have much stronger founder events, many of them with more than 1 million people. And it's a huge opportunity for medical genetics in India, which is not really recognized. There's up to 100,000 predicted new disease cases of newborn births a year from this process, and it could be averted or controlled or addressed through prenatal testing and pre-marriage counseling of the sort that's regularly applied in Jewish groups, in Finnish groups, in Sardinian groups, in Hutterite groups, and Amish groups in the West. And it's not really recognized in South Asia as a major opportunity. So in summary, ancient DNA is teaching us humility, that much of what we thought we knew about the past is wrong. There are many, many examples. I just showed you 3. That we're all mixed, not just from the times of Denisovans, but continuing to today, that no one is pure. I think it shakes us from orthodoxies that we've come to believe and learned when we were growing up, and that we learn even from scholarship in other fields, including our own. And it shows us how we're connected to each other in myriad ways that we had not previously imagined and usually cannot guess. What's next? I think what's next is applying this new scientific instrument to all times and places. This field is so new that it's extremely unequal in distribution around the world where the tool has been applied so far. Very European focused, very little focus on the Americas or Africa or Southeast Asia and East Asia, and that needs to be corrected. And even within Europe, and of course in many of these other places, many key time periods are not yet covered. Another important area is to realize the promise to reveal as much about biology as about history. In order to learn about biological change over time, you have to be able to watch the frequencies of these genetic mutations that affect biology change over time. And you can't do that with 1 or 2 or 5 samples. You need hundreds of samples. But that's what we're now getting with this rapid increase by a factor of 100 in just 5 years. And it will continue to go up very dramatically. It's absolutely clear that within a few years, we'll have many tens of thousands of ancient genome sequences. I think what's really important also is that as genetics begins to make statements and result in conclusions and findings about other fields, we who are not schooled in anthropology and archaeology need to learn how to enter into equal partnerships with people in these fields and translate our work in a way that makes sense and can be integrated into these other disciplines. And I think it's also important to do this work in a way that has respect for the ancient bones that are from people who are related to other people, past and present, and to treat the remains with respect. So thanks.
So, be delighted to take some questions and answers. There's a question over here. While the microphone is getting there, I might ask a question just to start. So, present-day contemporary humans have about 1% Neanderthal DNA. If you look at your 40,000-year-old sample and ask, can you find small fragments of Neanderthal DNA in addition to the long segments that suggest recent ancestry? Are there smaller segments suggesting more remote admixture that might help time the first encounter between humans and Neanderthals?
Yes. So, if we go there. Yes, so actually in this individual in Romania, you see these 7 large regions, but you also see shorter fragments. So presumably this individual has the contribution earlier on in his ancestry, probably in the Middle East, and then this recent family member.
And can you time that?
So the best timing of this early contribution is in the order of 60,000, 70,000 years ago. And then this sort of more recent one is within 6 generations. And we actually sort of confidentially have 3 early modern humans now also in southeastern Europe that are quite similar to this individual. It seems that the earliest modern humans really interacted a lot with Neanderthals.
Yes, over here. So for both of your presentations and thinking about past and modern mixtures like Neanderthals versus current day populations, that has begged the question a lot about what is considered a species in hominids, and whether that question can be answered by looking at divergence rates of the genome between Neanderthals and Denisovans and other ancient humans versus modern ones. And so my question is more specifically, in current day mixtures of very distant populations that are living today, are their genetic divergent as humans and Neanderthals or less?
So Neanderthals are more distant from any present-day humans than any of us are from each other, first of all. And then sort of this other question about what is a species or a subspecies, I generally totally sort of avoid the question by saying that's a sort of a totally sterile academic discussion. You can have sort of different definitions of species. So one definition would be, can 2 groups have fertile offspring, for example? Then clearly Neanderthals and modern humans were the same species because at least to some extent one had a fertile offspring. But of course, with that definition, then sort of polar bears and grizzlies are the same species because when they meet in the wild, they have fertile offspring. Then we would say polar bears are not becoming extinct because there are grizzlies around, which would be totally ridiculous. They are adapted to different environments, behave differently, look differently. So, if we describe the biology and the behavior of these groups, that's an interesting thing. If then these academic professors want to call them species or not, I don't care.
So this is for David, the remarkably successful Yamnaya invasion with their horses and their carts, when they came in, were they a genetically homogeneous group, or were they themselves composed of groups that you could differentiate between?
The question is about the Yamnaya and how homogeneous were they genetically across their range associated with their culture, material culture. And the answer is that they are remarkably genetically similar to each other across their range. We have data from quite a lot of them from different places now, and there are subtle, significant differences, but quite similar across their range. They themselves are a mixture of earlier groups, but they had homogenized over that time. That's not true of every ancient material culture. So it's tempting to think that there's a one-to-one mapping of the way people made pottery or buried their dead with ancestry. But for example, another group in Western Europe that we've studied pretty intensively, the Bell Beaker culture, which originated probably from Iberia, actually is genetically very inhomogeneous. It seems to be that it spread from Iberia with people of just typical European farmer ancestry and spread into Europe with people who copied that way of making pots and other traditions of burial, but into people with steppe ancestry, and it was practiced simultaneously by those two groups. So it's not a simple one-to-one mapping in any way between ancestry and culture.
So if I can follow up on that. So this gives me, naively thinking about it, the impression that there was a sudden event that established the Yamnaya success, let's say horses plus the wheel, whatever, and then a small population exploded. But that would suggest that there might have been considerable inbreeding, like Sardinia. Do you have any evidence for that, that they were, inbred, that there was a strong founder, in fact, in that population, for instance?
We don't have enough resolution with the data that we've been able to collect to determine whether there was a strong founder event in any of these populations. It's very clear that we will be able to in the next couple of years. That's one of the exciting areas. So I think we'll learn a lot about how these groups spread. There's interesting, it could be technology, most likely it's just some cultural package they had that helped them to spread. But it is also clear, and it's been shown in ancient DNA, that many of these ancient individuals from this time died of Yersinia pestis, the agent of the Black Death. So about 5 to 10% of these ancient steppe individuals had it in their teeth, which meant they were bacteremic at the time they died, and that means they probably died of it. So it's clear there were plagues endemic on the steppe. And one interesting speculation, which is not validated yet, but there's quite an increasing amount of evidence that is helping to push in this direction, although it's probably wrong, is that the people on the steppe might have been immune to certain diseases. For example, pneumonic plague, which would have been coded for by this Yersinia pestis at the time, which lacked the virulence factors for bubonic plague, flea-driven transmission. And that maybe the European population was naive, just like Native Americans were naive for the diseases that Europeans brought later on.
And that's why all those males bit the dust, not because the females preferred the incoming new guys.
We don't know. We just don't know.
Excuse me, there's a question up there in the corner.
Yeah.
Joel Cohen has a question.
Thank you.
Thanks to both speakers for the privilege of hearing these presentations. In one of David Reich's slides, there were outliers from South Asia, and it occurred to me to ask whether they might have been Roma or equivalent to Roma, and I'm wondering whether you have looked at Roma genetics in relation to the story you're telling, or whether it's completely the wrong time scale. A second subsidiary question for both speakers is, how can scientists help get the word out about the results of your studies?
So the first question is about the Roma. So this is the so-called gypsy population of Europe, that uses a language of South Asian origin. And genetically, in other studies, we and others have studied the genetics, and they look like a mixture of European ancestry and South Asian ancestry. It's clearly that mixed in medieval times, clearly some South Asian population, it seems from the genetic data, likely from the northwestern part of South Asia, made it in some kind of journey into Europe and dispersed across different parts of Europe. I think that it's really important to try to communicate these types of results to people, other scholars in other disciplines who work at impacts, as well as to the public, to scholars in other disciplines like archaeology and anthropology, linguistics and history, and to members of the public whose ideas about the past it might help to contextualize. And I, for example, wrote a book last year that was aimed to try to translate for those communities, because people kept asking me, especially my scholarly colleagues from these other disciplines, how to, what these results meant, and our papers are cryptic, and so I felt that that was an important part of what we had to do as geneticists. Svante may have something to add.
Not really, no.
The book is Who We Are and How We Got Here. It's a terrific book. It's beautifully written. question, Fernando?
I have two questions, really. One of them has to do with the migration of people out of Africa that is always presented in a one-way direction. And it always has puzzled me that if you have a passage that allows people to move one way, surely there must be times when people move the other way as well. And yet you find no genetic evidence of that. The second question has to do, you clearly have examples where you think a genome clearly belongs to Denisovans or to Neanderthals or modern humans. Do you ever find cases, encounter examples where you're just not sure how to place it? And what is your criterion to decide if this is a variant, say, from modern man, or is really a Denisovan or a Neanderthal?
I'll take the first question. So I think the answer is that people have moved back and forth. It's clear that the deep roots of our own ancestors have been in Africa at certain times in the past, and that's not from the genetic data for the most part, but mostly from the fossil record. So early upright walking humans with many of the adaptations we have prior to about 2 million years ago are all in Africa so far, as people have seen. But after 2 million years ago, Homo is both inside and outside of Africa with skeletons in Georgia and Indonesia and China, as well as in parts of Africa. And at this point, it's not clear where most of our own ancestors sojourned between about 2 million years ago and about 500,000 years ago. It's most likely, based on certain cultural evidence, that it would have been East Africa, but it could very well have been parts of Eurasia, which is a rich and diverse continent, which certainly could have supported complex evolution. After 500,000 years ago or so, it's very clear that our own primary lineage moved back into Africa or was always there. And modern humans evolved in Africa between 300 and 200,000 years ago, at least, as documented by the skeletal data, and then increasing amounts of genetic data after that point, and then moved out of Africa and the Near East after about 50,000 years ago again. But the story doesn't end there. So, for example, there are repeated movements back, especially from Western Eurasia into Africa. For example, Khoisan of southern Africa have maybe 5 or 10% ancestry from the ancient Near East, we think, due to the spread of herding into Africa, a technology that originated in the Near East and was brought by a pastoralist group that spread over that region, East Africa, beginning 3,000 or 4,000 years ago, and that made it to southern Africa. There's little bits of Western Eurasian ancestry, even in Nigeria, and there's some evidence that the proportions are even higher, although I think that's contentious right now recently. So there is spread both ways, but it's very clear that the primary ancestry of modern humans outside and inside of Africa prior to 100,000 years ago and after 500,000 years ago was in Africa itself.
Another question was Neanderthals and Denisovans, and if we find things that are intermediate more. So it's of course always a definition. I think one could have called Denisovans a sort of Asian Neanderthals if one like. But the sort of divergence is deeper between them than between any present-day people, and so we sort of choose to call them a different group. But we are really careful in not giving them a species name again. It's sort of just a trivial name. We do find mixtures in the sense that we find this individual now who is actually a first-generation offspring, and we find a bit of gene flow. So far, we've only documented gene flow from Neanderthals into Denisovans, actually. Probably we will find it going the other way, too. And I think we are, in a sense, lucky that this Nisswa Cave is probably in a zone where these two groups met. And that if when we look further west, we don't see Denisovan remains. When one will now get in the future more information from China and from other parts of Asia, it may even be that we find other groups actually that we don't know about yet.
Take one more question.
Thank you both for a wonderful talk. One of the most fascinating bits of data that you showed were how certain immune genes, like the toll-like receptors, are shaped by Neanderthal ancestry. And so I was wondering, are there other immune genes where we detect these associations, such as those that are particularly important for response to infection, like the HLA locus, for example?
So there is some evidence for MHC alleles coming over from Neanderthals and getting to high frequency. There's some claim that some Denisovan versions also come over. And in most of these cases, there are sort of two groups of genes where one sees things that have risen to high frequency as if they were positively selected. One group are immune-related genes, and for some reason, the other group are things that have to do with pigmentation and skin for some reason.
Well, I'd like to once again thank David and Svante for spectacular presentations and congratulate them on this year's Wiley Prize, and thank you for your participation.